Uh, so for those of you who joined in late, we are doing these webinars as part of DataFest 2017. Just 1 minute, I'll put this webinar live on Facebook as well. Just give me a minute. Right. So hi and welcome to this webinar on information discovery in life sciences. We have our speaker Vatsal with us as well. Uh, just to set the agenda, as I said, this webinar is happening as part of DataFest 2017, which started on 1st of April, coming close to its close on 30th of April 2017. As part of this, we've done various events, including we have done various competitions. We have done various meetups. We started doing these webinars. So I hope you have been part of it. Various prizes which are up for grabs, starting from various machine learning hackathons to skill tests. The last machine learning hackathon happens on 30th of this month. A couple of things which I just wanted to announce, and then I'll hand it over to Vatsal. So we would later this year we would be coming to Bangalore for a large conference which is called Data Hack Summit 2017. The idea behind this conference is to get the advanced analytics audience together in a room in a 3-day format. We'll do various workshops, so there would be 4+ workshops, more than 30 talks, various startup tracks. So the website for Data Hack Summit is live now, so you can go. So we'll start putting more details there. And as I said, the idea is to kind of create an opportunity for data scientists to meet with other data scientists in the community. The second thing which we are starting is data hack camps, which are like boot camp style learning. These would again go live later this year. And the idea is to do hands-on problem solving with industry experts and mentorship. So with those two, we'll kind of come to the webinar for today's session. The speaker for the day is Vatsal. So Vatsal is part of the CTO office, and then he leads the effort at CTO office at Ennoplexis. He brings immense experience in life sciences and domain experience on both life sciences side as well as data science side. And then one of the most wonderful people to kind of listen to. Their product, iPlexus, gets used across academia as well as industry. And I'd like to welcome Vatsal to the session. Let me just bring him as a panelist. Just give me a minute. Hi, Vatsal. Welcome for the webinar. And thanks for taking time out for doing this session. Really pleasure to have you as part of this webinar series. So I'll hand over the control to you, and then you can start taking the session from your end.
Yeah, thanks, Kunal. So I hope I am audible clearly.
Yes, you are. Thank you.
Okay. So thanks everyone, and thanks Kunal for giving me this opportunity. Um, so Kunal already gave a good introduction about me. So I'll start with very basics about what we do at InnoPlexus. So the idea behind InnoPlexus is providing the continuous analytics or information to the users. So I'll come down to how it helps the life sciences in next couple of slides. But to begin with, the major problem is that still a lot of analytics today is batch processes. You get the data, you process it, and you give it to the clients in form of some graphs or something. There lies the biggest problem. There's a lag in the information that client gets, and you are repeatedly doing the same things. Data scientists should be doing newer and fun things rather than repeating the same stuff. So this is what InnoPlexus is based at. We collect all the data, put it in a data as a service platform, and build applications on top of it. So we'll cover some of these use cases today. So I'll first, Kunal, maybe we can run a poll, say asking people how many are comfortable with life sciences. Do they really understand life sciences, or to what extent?
Sure, just give me a minute. I'll look. I've just launched a poll, which you would be seeing on your screen. So you can start voting at your end, and we can, so that Vatsal can have a. Great. Thanks everyone. So Vatsal, here are the results. So you can see. So just above half of the audience doesn't have background in data science. Sorry, in life sciences. About 32% of them know the basics, and 12% of them are saying that they have been in industry for some time.
Okay, so it is as I expected. A majority of you don't even know what basic life sciences is. So you all must have studied biology in college and everything, but that's not really life sciences itself. The main aim of life sciences, in my opinion, would be improving the health of diseased people. Some sort of disease you everybody has every now and then. So how do you cure that and then maintain human health? There are day-to-day things that you are providing through, be it food or nutrition, be it a basic consumer health care. So there are a lot more things into life sciences. So I have put some of the major categories. By no means this is exhaustive, but when we talk about life sciences, the majority of the money goes into disease diagnosis and developing drugs. So everybody has disease. You go to a doctor and he says you have this disease, but he needs to have that data backing him up to say you actually have this problem. For simpler problems, it's just he can ask you a few questions and then answer. Now, for more than that, you need machines and you need softwares to measure those factors, be it blood test, be it different kinds of EEG, ECG, and different graphical information, as well as then making sense out of it, which usually is done by doctors till now. A lot of research is going into automating that process, assisting doctors in making a decision about what this disease could be and how it could be treated. Then developing drugs, everybody knows those little tablets you take, but there is usually 10 to 15 year lag in developing that drug. And in those 10 to 15 years, it's not only discovering a molecule which can actually cure, it's the process of development itself. You have to try it on first a small animal, then on humans, a small sample set, then large human set, and finally launching into the market and again taking notes from the industry. So basically, this is a repetitive process for each drug taking a large amount of time. Because of this time, the drugs gets delayed and patients don't get their solutions. Coming to it on the next slide. But before that, other things you might have heard of making devices, like now you see fancy devices at hospitals, be it MRI, be it CT scans. Then there are newer and smaller devices to measure your insulin on a daily level, your sugar level on a daily level. And other side is consumer healthcare, your day-to-day products, from napkins to sanitary products to all the healthcare products. Then food and nutrition, and trackers and softwares are newer software that you see. It could include your Apple Watch, which notes down your heartbeat, your softwares in your phones, which track your health while you are running, you are exercising, and so on. So it's a growing market, but still the majority is treating the disease for patients. Now, in that case, the biggest problem we see is there is too much data. Problem is not the data these days. You are getting the results or records for every single patient, for tons and tons of research, for experiments. All the data is out there. And surprisingly, a lot of them is online as well. You can just go online and see a human genome itself, which is sequence of your DNA. You can see all the experiments in humans, in animals. All the research is publicly available, and still it's not giving insights to doctors. Because when somebody comes to a doctor or a researcher developing a new drug, they just can't have the right piece of information whenever they want. And the result, patients don't get their treatment on time. Now, how do we resolve it? Obviously, the bottleneck is converting the data into insights. And this is what I'm going to cover today in terms of information discovery. So what are the information sets that are available in data science, and how do we solve them? So first, I'll cover the different problems. Why is nobody working in life sciences today, or why are there so few industries that target life sciences for analytics? A lot of you, if you are a part of data science already, you would know majority of research is one into financial domain and two in retail and consumer markets. And why is that? Because all of them have structured data. 90% of the data analyzed in financial and retail companies is structured. You go to Amazon, you order something, that record is very, very structured. It has your username, it has timestamp, it has the products you bought with their IDs, and what all you bought, when you bought. Everything is so structured that you can analyze using variety of tools, and you must be participating in this data fest. Problem with life sciences is almost all the data is unstructured. Any research paper, any news article, anything important is basically a text. Similarly, you have experimental results. You have these scans, you have plates. So there are biological experiments where they have assays they run in a machine, and they get these plates with pink lines or some lines which indicate how much strength of this connection is. In NMR, you get these complicated graphs of how visual some pattern is. Now, all of that is not really tables and rows or even classifier type of things that you deal with machine learning usually. So that's why life sciences is a much more complicated beast. I'll give a further example in problems is inconsistent nomenclature. In life sciences, you get the IDs of individuals. Oh, sorry, in retail, you get IDs of individuals who bought what. In life sciences, a same author would have published 10 articles, and almost all 10 will have different format of his name. My own articles, if you try to find, they are all with different names. Some say Vatsal Agarwal, some say Agarwal Vatsal, some say only Agarwal V. Now it's difficult for somebody to understand that is it the same Vatsal Agarwal we are talking about, or there are 10 different people called Agarwal V, and which paper belongs to whom. Another problem here within the same nomenclature is the genes themselves. So gene names are a lot of variety. Disease names are a lot of variety. So same disease, which we call, say, cancer in common language, is called oncology, or even more complicated, metastatic neoplasm. Now, all of them mean the same thing, but when you are doing analytics, you will have to group all of them together because you don't know what is talking about what. Otherwise, you will have three categories called cancer, oncology, and metastatic neoplasm. Next problem is too many dimensions. As we saw in the poll, a lot of people don't have the domain knowledge. Earning domain knowledge for life sciences is much more complicated than retail, insurance, and others, because there are limited dimensions, as we call them. So you have the person who is buying something, you have the properties of that policy or that object, and very few fields. In biology, there are unlimited dimensions, because frankly, we as humans don't even know half the properties we should be looking at. People thought genes are the final thing we would need. After genes, we would know everything. But as soon as we got into it, we saw there are so many properties within genes, and genes don't even contribute 1% of the entire human DNA. There is 99% other stuff going on. So every time there are new dimensions and new things that appear in life sciences. So that's why it's much more difficult to control. Then there are simple variations, synonyms, morphological forms. Like EGFR is a gene which is also called as ERBB2, HER2. Its full form is epidermal growth factor receptor. Again, similar to inconsistent nomenclature, but here it's just that you have different names for the same thing, and here it's more about the same thing called differently. MRI is known as magnetic resonance imaging. It's also a gene acronym. So there is a lot of mishmash of terminology in biology. Then let's see the kind of data sets. If you go into industry, a lot of people are targeting this data set itself. Some are also going into electronic health records, but majority insurance claims. Why? Because insurance claims are at least semi-structured. Semi-structured meaning patient demographics. It would be clearly written, this is a male of 45 years of age. This has this code of disease. These are the costs. This is the person who treated them. This is who referred them. So all that information is structured. Although it also has some unstructured information, which usually companies ignore or are not able to handle yet, are the comments. Doctor would have written a comment that this is the disease, but these are the special things to note. So this is a person who has kidney stone, but this is the dimension of that kidney stone. So there are unstructured information here as well. So a lot of companies target just the structured information to provide analytics, but we need to move ahead of that. Similarly, electronic health records are very much structured in some cases. Like you will have patient information, you will have his history, but unstructured information contains the scans themselves. Ultrasound, MRI, CT, you all must have at least heard of it, if not went through yourself. But these are not really easy to analyze. There are computer vision algorithms coming up, but very few companies actually target these. Then lab reports. So everybody gets this huge lab report, which uses so many terms which we don't even understand. But when you are trying to analyze, when you are trying to understand what doctors prescribe, how do they prescribe, what is the thought process, how should patients be treated in the future, how to make guidelines. All of that needs information from these lab test reports, which only a doctor sees once and you put in your cupboard only for the next time if you mistakenly have the same disease. Otherwise, nobody gets these lab reports. Now imagine if we can get all of this data out from 7 billion people on earth, the kind of massive data set we have. And companies do have these today. So there are a lot of companies which on all these big hospitals you see in India, Apollo and Fortis and so on, they have these systems. So all that data is recorded, but it's not utilized. Reason being, it's unstructured, there is no nomenclature, and it's difficult to process. Next is experimental data sets. So experimental data sets include, so this is fairly technical. Since the audience is not there, I'll go very briefly. So these are genetic experiments which contain genomics data. Then there are human DNA sequences and other organisms' DNA sequences that people are doing. RT-PCR. So these are just terminologies. You would hear these terminologies when you are working in industry about the data sets, and these are more or less on the biological side of things, not for treating patients, but building up new kinds of treatments for the patients. And finally, the published data, which I was talking about. So there are, if you believe me, about 2.7 crore research publications in the industry, which is out there. Everybody has access to them. Just go to PubMed and search for anything. Then there are millions of presentations, millions of PhD theses. All this data is publicly available, and we are not able to get insights from this. Clinical trials. For every drug you take, there have been plenty of clinical trials which are openly available. But it's not even used by most of the physicians as well as most of the researchers, because it's not easy to analyze as a whole. You can't really open 4 lakh trials one by one and check what's happening in there. And then news, patient forums, blogs, social media, which you are aware of. A lot of companies are trying to utilize this. There is a lot of noise in there, but still important information. So coming to the challenges that we saw and how to solve them. So this is the crux of it. What do we do with the standard nomenclature? One, understand the user query. So if user just types in Google today, non-small cell lung cancer, fine, a type of lung cancer, Google and most of the softwares, if you go to PubMed, they start looking for the terms. Non-small cell lung cancer will be treated as 5 different words, and it will look it up as the most likely combination of these words. So you will get results even for lung cancer, even though it's not really non-small cell lung cancer. So you need that standard nomenclature to understand the user query. On the second hand, finding the relevant information. So after understanding, you have to retrieve these results from, as I mentioned, 27 million publications, 2.7 crore, clinical trials, 4 lakhs, then congresses in millions and thesis in millions and news articles. You need to find the relevant articles, not just where it says non-small cell lung cancer, but it might also be written as NSCLC, the short form. It should also search for those. Similarly, it should search for all the synonyms in all types of articles. So how do you do that? First is, or the primary way to do it is build a meta thesaurus or an ontology. Now thesaurus you must all be knowing about. It's like a dictionary. Every word has a meaning. That's one of the parts. But again, what are its synonyms? Including synonyms, what are its short forms? Then beyond that, how is lung cancer related to non-small cell lung cancer? It's basically a parent-child relationship. and so on. So you have to build these relationships, these concepts, their synonyms and everything into one bucket. On a superficial level, how would you do it? You will choose the entity that interests you the most. Let's say first we are targeting just the disease names. Now collect that data with all the synonyms. So there are a lot of curated databases again that have this information, but is not usable for the exact purpose. So you can go to mesh ontology, you can go to a lot of sources to get that names of all the diseases in the world. There would be databases for ICD-10, which is a standard database of disease codes. So you get all that data, then map the connection between concepts. This is the trickier part. Now here, there are a lot of ways. One is whether that is somewhere published, some database which lists the relationship between them. There are some publications which highlight the representation between those connections. There might be sense within the words itself. Since lung cancer comes in non-lung sense, small cell lung cancer, you can tag that, okay, lung cancer is the parent for non-lung or non-small cell lung cancer. So you map these relationships, then store them in a graph database or an RDF format. So this is a standard for ontology storage, because you can't really store this kind of information in a relational database. You need NoSQL, that too specifically a graph data where you have networks of information. So I hope everybody, or at least most of you, understand what's a graph database. It's a node which contains a single concept and an edge which shows the relationship of two nodes. Similar to this structure you see on the left, this is one node, second node, and third node, and the relationship between these three. So this is the parent node, and these are the two child nodes. So you store the information in graph database so that when you search for one term, you can get relationship with other terms as well. Then you build an API where when you input a single concept, you can get the whole sets of information. One API could be give me the parent concept or give me a sibling. There could be another concept, another API, give me all the synonyms for this concept. So that if somebody searches for NSCLC, I get all its known synonyms and variations so that I can look it up much more effectively. Coming to the second challenge, which is so many unstructured data sources. So I am talking about ontologies. I am talking about publications, about news articles, social media. At all of those places, you have different formats. So you have same information given in multiple formats and multiple ways. So let's say we are talking about John Taylor. If I search John Taylor, I get these results: John H. Taylor, J. Taylor, Taylor John M, J. H. Taylor, Taylor James. Now, obviously, John M. Taylor and Taylor James cannot be same as John H. Taylor. So how do you identify whether they are the same people or they are the same different people, or how many different people lie in these seven names? Similar problem is there for the institute names. Delhi University is also called University of Delhi. Somewhere it would be written as one form, somewhere another form. On the other hand, there are IITs. There are 15 IITs. Every IIT has a city allocated. So there is IIT Delhi, IIT Bombay, IIT Kanpur, but they are actually different institutes. So how do you understand which institute a user is talking about in a publication, or what does this person want to search for? There are two ways. One is identity parameters, the obvious ones. Is the middle name same? If middle name is same, first name and last name are already same, there is a high likelihood this is the same person. is the age and specifically a date of birth same. So if you have some database where you have the date of birth available, like Twitter, it might be that you can access the date of birth of the individuals. In that case, you can say exactly this is the same person as a Facebook person who has the same date of birth. Email ID, again reliable. Then, although this information may not be much available, you have to go to the contextual parameters. Contextual means that individual has a lot of habits, had a lot of other things which are not concrete, but which can be designed using machine learning algorithms, like the writing styles. I am sure all of you had an experience in your life where the teacher saw the handwriting and recognized that this is not the same person. So everybody has a different handwriting, and not only handwriting, they have differences in the way they write. Everybody has a tendency to use few words more or less, specifically in the research domain. So they would be using different kinds of words. They would have different networks. So one co-author, author would have 10 co-authors, but 4 or 5 of them would be common in a lot of publications, because you have your friend circles, you have your researcher network, and a very close one. So not 2 people can have the same co-author network. And then affiliation. So which institute are you working in? Which institute did you study in? If you have those information, they can again be used. This, I was talking mostly about person name, but same is true for institute name. In institute name, again, you can have a lot of secondary features, which could be the address of that institute, which could be the website of that institute mentioned somewhere. So a lot of secondary parameters which you can take a guess from. Similarly, gene name or any biological entity, you will have one of the synonyms available. You will at least have its biological function somewhere coming out in the context. In the document, it would say that EGFR gene, mostly reliable for cancer treatment, does this. So you know cancer treatment is its function. So you can have these subtle hints which you can derive from the models. In entity recognition, now the example I just mentioned, you will have to identify these entities in the document first. That an EGFR is mentioned somewhere in 20 pages of publication. Which author is it talking about? So usually you are getting a bunch of documents. You are not getting the structured information, as in this is the author, this is the abstract, and this is, let's say, the title. But you get a what do I say, a text. Now you have to understand whether this is a person. Now how do you do that? Now there are name entity resolution or name entity recognition APIs already available, which pick pieces and predict whether it's a name or not. But more than that, you can enhance those further. You can use intra-word patterns. Like people from life sciences understanding would know if I just say CYR1 and ask you to guess what it is. I am sure people with life sciences background or understanding would know this could only be a gene. This is the way usually a gene is written. And this is an imaginary name. This is not an existing name. Even then, you are able to take a guess that this would be a gene name. So there are always patterns between the words. For example, can anybody really say this is a person name? Very, very unlikely. In no sense, this could be a person name. Maybe a Twitter handle, but not really a name. So you can build the models like LSTM, ANN models or RNN models where you can predict the introvert patterns. The second is the context itself. So usually when somebody would write, they would write gene CYR1 is responsible for this and this. So obviously gene word came immediately between CYR1. So you can take a hint that this would be a gene. But rather than making these rules, it's easier to learn from the data itself. You can build a very simple supervised model if you have few data trained, but even the unsupervised ones shouldn't be very different because there would be kind of words coming before and after it. And third is the easiest part, the context of the document itself. You won't find a gene name in any article related to, let's say, a sports website. They might name a disease. They might name the technologies, but they would never name a gene. So the likelihood itself decreases. So first you filter out the documents, then you apply intraword patterns, and finally you see the pattern of words before and after it. Third big challenge is the data size itself. As I mentioned, we are dealing with about 27 million publications and other records. All of that turns out to be about 100 terabytes on our end. Now, 100 terabytes of data, obviously there are technologies. Hadoop is there, Teradata is there, and so many other softwares are out there. But it's not about the software. It's about the data scientist, how you are thinking about these problems. I'll take an example which we faced a couple of weeks back when the data was only 26 million publications. Our day-to-day job was we get the data from a lot of sources. We had to tag 6 million known concepts, which is part of the ontology, into these 26 million publications whenever there is a smallest change. So we changed the format of publications to something, and we improved the algorithm a little bit, and we want to tag again. Now 26 million publications, in each of them, at least say 5 to 10 pages, we have to search all of 6 million concepts. And this happened almost every day. So 156 trillion loops, which runs for 5 days, and we need to run it every day. We had indexed all the publications. We used Redis to store the concepts. We optimized the input output, building everything into a single network. We used 5 machines for parallel programming on 4 cores. 4 cores each machine, that is 20 cores. And still it took 5 days after all that. Before it used to take a month or so. Now we thought, okay, let's move to the Spark, which is a Hadoop technology, quite a popular one. And the two problems we ran into were, first, too many partitions. We had 26 million publications. We set up 3 systems with 64 cores each, which is like 200 cores. And we tried to divide this into, I don't remember exactly, but like 100,000 publications in each partition. Lesson learned, you should have only 2 to 5 times the number of cores available. So once we reduced the partitions to about 500, it ran perfectly, but we had too many partitions earlier on. So however big the data is, partitions should be limited to this number. Another problem was slow processing, because Spark stores the data in memory. Everybody would be aware. Now, if you store these 6 million concepts, I think this was about 50 GB data or something. Now 50 GB data, no, sorry, the original data was 300 GB. If you try to store 300 GB in data in the memory itself, obviously, even in multiple systems, that was not possible. But Spark had to use it in every core, or every machine rather. So in that case, that was definitely not available. It dumped everything into hard disk, and everybody knows reading and writing from hard disk is about 100 times or 1000 times slower than in memory. So we were back to the square one. It was taking even more time than the earlier. One of the techniques I came across and might help a lot of you is, when you are tagging, you might use hashing. So instead of using the whole concept with everything, we just used the hash. Hash is kind of an encrypted code, but you can limit it to, say, 64 bits only. So it's usually used in encryption and passwords that you are using. So all the passwords are encrypted and stored safely. Here, safety is not a concern, data reduction is a concern. So if you have 64 bytes and any concept could be stored in those 64 bytes, that solves our problem. Instead of taking, 64 bits. So instead of taking 300 GB, this got reduced to 30 GB. Because everything is now only 8 bytes. So these were the information discovery challenges we had. After information discovery, so we enabled the discovery. Next part comes is intelligence generation. And since we are lacking time, I'll cover slightly less depth in intelligence generation. So first use case we have is the search results themselves. Everybody uses Google search, and at one point in the beginning, you would have noticed it says something like 15 quintillion documents found or 3 million documents found. What do I do of that number? That number is useless, right? Any number of results you find is not going to help me. I am only interested at most on top 20 results. And those 20 results may not be absolutely my focus. What I will have to do is I'll have to think, what should I search next? What would serve my purpose better? Wouldn't it be helpful if the search results themselves tell you what kind of concepts are in there? What are the popular searches? What are the upcoming trends here? Some insight which helps me derive my next query. Help me find the right query that I want to search for. So users want knowledge and not the data. Currently, search engines dump the data, not provide any knowledge into it. What you can do is you can summarize those results. Top concepts that are present could be simply by frequency. Which concept came most, which came second most, and so on. But better would be over-representation. Over-representation is out of 1,000 documents, only 20 talked about lung cancer. But when I search for something, maybe EGFR, I get 50 documents, out of which 15 are now lung cancer. So out of 1,000, I only had 20, but out of 50, I have 15, 1-5. So my coverage increased from 2% to 30%. So lung cancer is overrepresented in case of EGFR. So there are a lot of algorithms that can be used for overrepresentation, but yeah, you can simply calculate it yourself. The biggest problem calculating yourself is if there is only one article. In 27 million, if there is only one article for some topic, I search for something, I get 5,000 results, and that one article is still in there in those 5,000. I actually have 5,000 times over representation, which is incorrect. That article itself is not relevant, it just got represented in this data set. So there are more algorithms like there's Google's GMD, which calculates taking into factor how much important that article was earlier on. So you can look into those further. Second, more interesting thing would be, especially for a life science or a research domain, would be upcoming topics. I know lung cancer would be overrepresented in EGFR because it's being studied for 25 years. Tell me in EGFR what has happened over last 1 year. So two ways again. First is you can simply have overrepresentation in a time-bound fashion. You can take entire data when searching for EGFR as your background, and how much is it overrepresented in last 1 year. The second could be the trends. Is that concept rising in the last 5 years? You can just do a time series analysis. Second is document similarity. Again, a big problem in information discovery is I found a jackpot article. This really helps me what I want to do. Can I see 10 more articles? Nope. You will have to search all over again, look through 10,000 documents and figure out if the second one is of use to you or not. wouldn't it be helpful if I search for just one document and see all the similar ones? How do you do it? Simply take a document, separate the structured fields, if any, run entity recognition we discussed earlier on the unstructured fields, and extract the concepts being talked about. Out of those concepts, build a network. Which ones are most important? So TF-IDF, I am not sure if everybody is aware of. It's just the frequency of that word against the document frequency. How many times? For example, the word THE, the, would appear in almost all the documents. So its important is much less than lung cancer. So find the important concepts and build a relationship between them based on are they in close proximity? Do they have existing relationship between themselves, and so on? And then run this over all the documents you have, 27 million publications, news articles, and social media, etc., and see which have the similar network of concepts. What do you have to match there? The most common topics. Are they occurring there with a similar frequency? Is the relationship between the concepts similar? So two words occurring nearby, and so on. And you get the documents which are very, very similar to the original document, which serves a big purpose of information discovery. And my final use case would be the top KOLs. So within the network of humanity, who are the top influencers? So it's little bit used in other industries as well, but it's very, very relevant in life sciences or pharma domain, where you want to find a scientist who can help you develop a new drug. Now, how do you find such people? The traditional way is you just go find a few articles, see who published them, talk to them. Second way people do is citation analysis, which means who are the people which get most referred. So there are few articles published in 1970s and 1930s, which is like gold mine. People still refer them. Similarly, there would be some articles which are referred very frequently. People say these are the top guys because they are referred most. Third is surveys. Go and ask people who they think are the best. But not really helpful because these methods are very, very biased and subjective. Easiest way is build a social network yourself. You know people on who are friends on Facebook, who are friends on Twitter, or in publications who are all connected to each other. They're co-authors, entire network. Find the central nodes. Now there is the concept of network centrality. Centrality means not just highest number of connections. Somebody having 500 connections may not be important. Why? Because they know 500 people who don't know anybody at all. On the other hand, somebody knows 20 people who know 1,000 people each. So there are different ways to calculate the centrality of a network. So these are the 5 nodes which are very, very central. Again, it's use case dependent. If you want to find somebody to spread the information, say when you are doing marketing, you would identify people who would be able to large number of people themselves. Whereas in terms of research, you would want people who have a really good network of researchers. They may not know many people, but they know 5 people who are very good researchers themselves. So that person can help you develop a new drug much faster. So different centrality measures for different use cases. Then another variation to this is emerging KOLs. So the top guys are probably 50, 60 years old. They're already working with 10 companies and a lot of projects, very busy. And they wouldn't be top 5 years down the line. You want to target people who are upcoming these days. How do you do that? That's the tough part, because there is no traditional way. You can't find the highest citations because they are already top KOLs. Surveys won't help. There won't be any people who know the guys who are emerging in the true sense. What they do is, okay, these are my personal connections. He's doing really good these days. You talk to him. Again, social network analysis is helpful. You have the network ready. You see the increase in their centrality or connectivity. Okay, in 2012, this guy had 10 contacts who had 15 contacts each. In 2016, that guy had 100 contacts, 20 of them had 1,000 connections each. So this guy is really growing. Then you can set again a minimum threshold similar to the overrepresentation case. Somebody who only had one connection in 2012 and who has 15 connections today has grown 15 times, versus somebody who had 10 connections and now has 100 connections has only grown 10 times. So it's not really the purpose of just seeing the multiplication factor, but really the growth. So there are again chi-square distributions and other distributions which give you an idea about who is really increasing. So this is all I had in terms of the content I prepared. And for the demo, you guys can check iplexus.co. It's freely available for personal use. Only for commercial use, you need the license. And then you can try out these different keywords and see how it identifies lung cancer as a single concept instead of two words. It identifies Erbitux and shows you a detailed view. These are the results summary I was talking about. So for Erbitux lung cancer, you see malignant neoplasm of lung as the first result. Small cell, non-small cell lung cancer as second result. So these are relevant topics in this context. So you can check it out, and that's all from my end. Thanks a lot for hearing, and I would like to take questions if you guys have any.
Sure. Thanks, Vatsal. Very informative session. In fact, it just reminded me of an article I was actually reading a few days back, which said that I mean, a lot of researchers kind of estimate that we only know about 3% of our brain, or we only understand what our brain does to about 3%. So a lot of things in that sense are still being discovered in life sciences. So let me, kind of pick up some of the questions which have come up. So the first question is from a person who says that, given that most of the problems are, have a really large magnitude and then are difficult to solve, are typically project life cycles in life sciences longer, or how different they are versus, let's say, a banking or some of the other domains? So are they all long-term projects, or are there some short-term projects as well?
Okay, so I would say as such, there are all sizes of projects, like in any industry. What you see in financial domain today is as a result of last 10 years of maturity, that they have a lot of algorithms pre-built. Therefore, you see the small projects where you run the pre-built module, RFM analysis, or some outlier analysis, which you can directly apply to the data. But in life sciences, there are very few pre-built algorithms. However, as such, the human knowledge is incremental. You only see a small part of the problem that you are trying to solve. So as such, projects at InnoPlexus and a lot of companies are generally 3 to 6 months. The picture I showed you is the holy grail, I would say, the end goal. But in terms of projects, we don't take more than 3 to 6 months, have a concrete deliverable, what user would get at the end of 3 months, and keep repeating the cycle for, let's say, 20 years. That's not a problem.
Got it. Thanks also for that. The next question is from Karthikeyan, and he's asking that can you brief about the technique used for concept tagging? Is it topic modeling, and how do you measure and validate the accuracy of tagged results?
Right. So a very good question, Karthike. We have spent like years on making that concrete, and I wouldn't say we have the 100% solution, but obviously the first and obvious solution was just keyword tagging. Just look up that keyword. If it's present, that's fine. Then you have basic checks like is it capital, small, the case, then special character handling, and so many other things. Topic modeling came midway, where you can just find what topics it's talking about. But mostly you need to have an ontology pre-built. You need to have not even an entire ontology, but you need to have database of concepts that what terms are usually used or what terms are about life sciences that are present. And then you can do basic LSTM or RNN model to just look if this is very similar to an existing concept.
Sure.
So I think that's where I will stop, because I can go on for hours about topic modeling itself.
Sure. Okay. Okay. And as a follow-up, he's asking that can the creation of ontology be automated to some extent? And are
So
there
I would
tools
recommend, as an individual project or as a small project, you should use the pre-built ontology. So you can find disease ontology, gene ontology, GO and DO. They are publicly available. Just download that and use that.
Okay, great. In fact, that kind of leads us to the next question. So the next question is that if someone wants to kind of access some of the open data sets which are available, so the first question is, are there some good open data sets available? And if yes, what are some of them which you would recommend for a person to start?
Right. So ontology, I already mentioned DO and GO. And for publication data sets, go to PubMed directly. So they provide you the entire data for everything 27 million. If you can download that and use it, it's fine. But you can select small pieces. Then there would be other sorts of dumps available at a lot of places. You can clinical trials won't be that easy. Congresses are again tough. Thesis databases are publicly available again. You can get thesis from a lot of universities which provide you the dumps. But yeah, start with PubMed. Go with the Medline data.
Okay, okay. Thanks. The next question is actually from a beginner, that how can a beginner, a college student, start with life sciences? He says that he feels that it's pretty intimidating for a beginner. So how can a person who is a fresher can get started in life sciences and data science?
Yes, I would confess here that it's not only intimidating for a beginner, it's still intimidating for me and a lot of top guys I know. So the first thing is, don't worry about it too much. Learn small bits at a time. Start with understanding the basics of biology. I know it's scary for a lot of people in engineering background and analytics background, but just go through the basic biology of things. Read a lot of news articles, be it Times of India or New York Times or anything, where there is information about life sciences. If you read about stuff, you at least can connect the dots, that what I'm doing, what pieces are connected, what user actually wants. The biggest challenge usually people have in life sciences domain, even the expert data scientists, is they don't understand the context of what they are doing. They are making perfect models, but they are not able to understand what they are doing because they lack that basic knowledge. So first thing, go to the newspapers, read life sciences, what's cutting edge, what's happening these days. There was an article about a new company that Elon Musk launched, and there are several articles on that about brain reading and so on. So once you get into the touch of what things are happening, what can be done using life sciences, then pick a small data set, build small models, and then go on.
Sure, sure. Thanks. And any specific courses or any specific open material which is available, which people can kind of try and read or is more structured than the kind of open articles?
So that's the biggest blocker these days. Even in my education, there were no books, because for life sciences, it's more or less research dependent. So I'm sorry that I can't refer you to any book right now, because there are very few books, if at all, and they are all on the technical side of it, understanding the biology. You can start with a book like Leninger, which talks about biochemistry, but it's like 2,000-page thick, large book, which nobody wants to even start. So yeah, not a concise answer for your question.
Sure, sure. No worries. The next question is, how do you differentiate between identifying document similarity between supervised learning method and the network analysis method, which you talked about?
Okay, so as such, supervised algorithms don't work too well. So you have to build a supervised algorithm over some features, right? So what kind of features would you choose in a in document similarity for unstructured documents? You can only fetch the terms, their frequency, and so on. The problem there is, you can find the terms, you can find cancer in a lot of articles, but they are not about cancer. I have seen in my work a paper about detergent, or patent. There was a patent about detergent which was talking about cancer somewhere in between, but that's not clearly a cancer patent, right? So the problem is, if you use supervised algorithms, it will give you the similar terms or similar concepts, but they won't be talking about the same thing. Because if I say any medicine, let's say Brufen cures headache, if only this statement I start comparing with a lot of documents, what will happen is it will give me all the documents having Brufen and headache, but it won't give me documents which talk about Brufen treating headache. So you need that kind of network in there.
Sure. And then because we are kind of running out of time, I'll just take the last question. So this question is, the person is asking that from your perspective and your experience, which database did you find most scalable for doing graph-based, things which you talked about? And then specifically, the person is asking your recommendation on Neo4j as a database.
Okay. So you guys are asking a lot of right questions, because we went through all of this in last 1 year especially. So Neo4j, the biggest problem for us was the enterprise license is expensive. And the second problem, for some reason, for life sciences, okay, I don't want to come into bad books for Neo4j guys, but in my experience for life sciences, at least for our requirements, it's not really a good solution. It slows down, and the kind of network information you want to gather, Neo4j doesn't help you much. But as a beginner, it's an excellent resource. The Cypher query that they have can do a lot of things very fast. So you don't have to learn itself how to build a network, how to find the relevant information. So in a way, it's good. But again, if you want to try some of the things I mentioned, like network centrality, in Neo4j, they don't have the solutions ready-made for a lot of centrality measures, and building it yourself is very complicated. We use ArangoDB because it provides us a lot of flexibility in terms of building our own model. And again, the enterprise license and everything is much more easier in ArangoDB for us.
So, how has been the scalability on ArangoDB?
Yeah, so ArangoDB is horizontally scalable. Neo4j, the biggest problem is the reads are scalable, writes are not. So sure, you get limited at certain point, but definitely not for personal use.
Sure. Okay. Great. Thanks a lot, Vatsal, for that session. I think again, one of the really good sessions, and which kind of gives a glimpse of a domain which doesn't get talked about as much, and I think it's probably the domain which kind of makes the most impact. And in fact, the 3% statistic which I was talking about came from the Neuralink article from wait,
Yeah,
but why is
I,
so hoping to see some really good innovation, and then I'm sure the domain would kind of become very interesting in the years to come, given that people like Elon Musk are kind of seeing
yeah,
opportunities here. So
yeah.
thanks a lot for taking this time, and then I thoroughly enjoyed the session. I'm sure the community enjoyed as well. So thanks a lot for your time.
Yeah. So thanks for the opportunity, and thanks for the audience for patiently listening and with their intelligent questions. Thanks a lot, you guys.